So hello class, as I discussed earlier, so the today's presentation is based on recombinant DNA technology, specifically methods of gene transfer, right? So earlier we have seen the different molecular tools used in the recombinant DNA technology. Then we have seen the different type of vectors, right? In today's session, we will study the different methods of gene transfer. So now this gene transfer method means what? Actually, introducing foreign DNA to the cell is a very difficult task in the biotechnology. But utilizing some natural gene transfer methods and by some laboratory methods, we can perform this transfer of recombinant DNA, right? So this gene transfer methods are specifically involved here. The first one is the transformation. So we have seen it in the bacterial recombination earlier. Conjugation, the physical transfer of genetic material from the donor cell to the recipient cell. Transduction. So these are first three are the natural processes of genetic transfer between the bacteria. The next one is electroporation, where the electric pulses and high voltage is used to fuse that particular DNA fragment, or we can say to increase the permeation of cell membrane. Lipofaction, where the liposomes are used as a carrier for encapsulating the DNA. And the last one is direct transfer DNA. By using microinjection or by using particle bombardment, we can directly insert the DNA fragment to the recipient cell. So these are the 6 gene transfer methods we have to see in detail. Now, what is transformation? As we have studied earlier, so the transformation occurs or it is associated with the transfer of the bacterial gene from one to the other when there is a depletion in the nutrient medium, right? So that time that the bacterial cell goes to the lysis, and after lysis it will release the DNA or the fragments of DNA which get attached with the other bacteria, and that it get combined with the other bacterial chromosome, and it will form the recombinant DNA there. So it is natural process. We can carry out this process in the laboratory by carrying out the lysis of the bacterial cell. Now, how to introduce this foreign DNA? So that the introducing the foreign DNA into the bacterial cells, and how this introduction or uptake of plasmid DNA by the recipient cell is carried out, right? So we are studying here how the recipient cell will uptake the plasmid DNA from other microorganism or other bacteria. So generally this uptake is carried out by cooling followed by subsequent heat shock. So initially we treat it with the ice cold calcium chloride solution at 0 to 5 degrees Celsius, and subsequently heat shock at 37 to 45 degrees Celsius for about 90 seconds. So because of this, that particular plasmids get uptaken by Escherichia coli. Now this transformation frequency get varied with the bacteria to bacteria. The transformation frequency which transfers to the fraction of cell population that can be transferred. So we are not transferring the complete one, we are transferring the fraction of cell population, and it is reasonably good. So this is actually we are performing into the transformation, right? Initially we are treating that particular recipient cell at ice cold calcium chloride solution and subsequently heat shock condition. So because of it, it will going to like we can say the cell wall get produce the permeation part, and because of that permeation, that plasmid DNA get inside the Escherichia coli. So as it again happens in case of now how it is happening outside the, or how it is happening in the laboratory. So at the one side you can see the bacterium. So the bacterium is having a plasmid, and it is also carrying the antibiotic resistant gene. On other hand, the plant cell is there with the particular plant DNA. So here the plasmid comes in contact with the particular plant cell DNA, and further with the help of DNA ligase, it is forming a plasmid plus plant DNA. So it is happening when the bacteria goes to the lysis. So it is again the transformation method. Now the next is transformation efficiency. Now what is transformation efficiency? The number of transformants per microgram of added DNA. हमने जितना डीएनए ऐड किया है उसमें से कितने ट्रांसफॉर्मेंट्स है पर माइक्रोग्राम ऑफ एडेड डीएनए. So the bacterial cells take up the DNA and are considered as competent. Now here the cell which is taking the DNA is called as competent. The one is donor, the another one is recipient. The one which is taking the DNA, that means the recipient, is called as competent. Now here the competence can be enhanced by altering the growth conditions. So we can alter the growth conditions, and by altering the growth conditions, right, we can further do the transfer. So now in this case, in this uptake, it is quite difficult to transfer or to uptake a large sized DNA, right? And that's why it happens only in case of small plasmids, and the size is generally 10 raised to power 7 to 8. Generally in case of Escherichia coli, the transformation efficiency is 10 raised to power 7 to the 10 raised to power 8 cells per microgram of the intact plasmid DNA, right? So it is believed that calcium chloride affects the cell wall and breaks at particular localized region, and it also responsible for binding of DNA to the cell surface, right? So this is actually the mechanism of calcium chloride. Again, instead of calcium chloride, we can use calcium phosphate. This calcium phosphate is referred for the transfer of DNA into cultured cell. Now here in this case, calcium phosphate is having some limitation. Now what are the limitations associated with it? So it precipitates, and as it precipitates, that's why it is toxic to the cells. So that is the reason we preferably go for calcium chloride. Again, the use of diethyl amino ethyl dextrin for DNA transfer is one of the chemical for studying the transformation efficiency. Now you can see that the conical flask is consisting of bacterial cell which we have cultivated in the laboratory. Further treatment with the chemical compounds, you can see the different chemical compounds are enlisted over here. It is producing what? It is producing the preparation of competent cell. Now the competent cell is ready to uptake the plasmid DNA, right? So again, it goes to the heat shock at 42. It is in the range of 37 to 45. So 42 degrees Celsius, and this competent cells further grow on a particular medium. So after growing, again we can check the transformation efficiency for the same, right? So this is the transformation efficiency. Now we have seen all about the transformation, where the breakdown of cell wall transfers the genetic material from the donor cell to the competent cell, that is the recipient cell. Okay. Now in case of conjugation, this is the another method of gene transfer. Here the particular what we can say that the cytoplasm get attached with the another one, that the donor cell transfer the genetic material to the recipient cell by F. pilus. So right, so F. pilus is responsible transfer of genetic material from one cell to the another cell. So here donor cell is consisting of a plasmid which is transferred to the recipient cell, and further that plasmid get combined or interact with the genetic it get exchanged with the genetic material of recipient cell and produce the recombinant DNA into the recipient cell. So this is again the natural method which we can use in the recombinant DNA technology for production of recombinant DNA in laboratory. Now a natural microbial recombination process, where the plasmid inserts DNA is transferred from one cell to another. Now one thing which is very different here, In case of conjugation, it may happen between two different genera. So Salmonella typhi belongs to one different genus and Shigella belongs to the another genus, right? So two different genera can exchange the genetic material by conjugation. So this is all about the conjugation, where the F pilus is involved for transfer of genetic material from one cell to the another cell. So in this method, the physical contact of the cell is necessary. The third method is the transduction. This is just a revision of the bacterial transduction we have studied earlier. So you can see the first image of bacteria or the bacterial virus, we can say, which was initially infected by the virus. So that virus formed its genetic material inside the bacterial cell. So in step 2, you can see the genetic material get fragmented between the pink lines and blue lines. So the pink lines belongs to the viral DNA and the green lines belongs to the bacterial DNA. Again, as it goes further, so that the virus is producing its own protein that is required for capsid formation and all. And in the third step, where the capsid or a complete bacteriophage is forming. So as the bacteriophage is forming, that capsid encapsulates some of the bacteriophages, encapsulate the bacterial genetic material, and some of them are carrying the viral genetic material. And as the concentration of bacteriophages increases inside the cell, as the base of the bacteriophage releases the lysozyme enzyme which dissolves the peptidoglycan, right? I repeat, the enzyme which dissolves the peptidoglycan is lysozyme. So as the concentration of lysozyme increases inside the bacterial cell, and on a third stage, you can see it releases the bacteriophages outside the cell. Now this bacteriophages, some of them are carrying the bacterial gene, some of them are carrying the viral gene, right? Now they transfer this genetic material to the another bacteria. So now the fourth image represents the recipient cell. Now here that bacteriophage get attached, right? Again, it penetrates. After penetration, it releases the genetic material. Now in a fifth image, you can see it is releasing the genetic material, that the blue line is associated with the genetic material of bacteria and the pink line is of virus. And in a sixth case, it is forming the new genetic material or the genetic material which resembles to the, or which is having the genetic information of both, right? So this is a recombinant DNA formed in the sixth image. Understood? Or sixth cell, we can say. So this is the recipient cell. So this is what all the bacterial transduction. Now the foreign DNA can be packed inside the animal virus. Now how we will transfer it? So what we can do, we can pack the foreign DNA inside the animal virus. So what we are doing, we are packing this foreign DNA to the animal virus, that is nothing but the bacteriophage, okay? And then we are using it as a carrier for the next one. So these viruses can naturally infect the cells and introduce DNA into the host cells. The transfer of DNA by this approach is called as transduction. Okay, so here the animal viruses are carrying this foreign DNA and transferring to the other cells. So this is called as transduction. Electroporation is the method which is based on principle, that is high voltage electric transfer. So in this case, we apply the electric field, that is electric field mediated membrane permeation, okay, and because of it leads to the cell membrane. That cell membrane going to fuse, and because of it exogenous DNA can easily enter inside the cell. So this electric shocks can induce cellular uptake of exogenous DNA from the suspending solution. Okay, exogenous means it is outside the cell or it is into the solution. So now that this DNA fragments, exogenous DNA fragments get fused with the bacterial cells, and it enters through the spores, and then DNA enters to the nucleus, and ultimately it is forming the recombinant DNA. So this is called as electroporation. Fine. Now the next is, as it is a simple and rapid technique, because it is very easy to take a DNA fragments and take a cell in a solution and to apply the electric field or high voltage electric pulses, which further fuse the cell membrane and allow the DNA fragment to come inside and form a recombinant DNA. It is simple and rapid technique, and it is an effective way to transfer Escherichia coli cells containing plasmids with inserts DNAs longer than 100 kb. In case of transformation, there were limitation. We were not able to transfer the larger plasmids, right? The plasmids which are of larger size, yeah, larger DNA was not allowed to transfer with the help of transformation method. But electroporation can allow the transfer of longer DNA to the cell. The next one is the liposomes mediated gene transfer. This liposome mediated gene transfer is associated with the liposome. Now, what are liposomes? These are the circular lipid molecules which have an aqueous interior that can carry the nucleic acid, right? So this is having the two layer. The outer one is the lipid molecule, lipid layer, and the inner one is the aqueous interior. So it also encapsulates the DNA in liposomes, and this liposome mediated gene transfer is also known as lipofaction. Now what happening in this lipofaction? You can see the liposome is there and the DNA fragments are there. When it makes, that liposomes encapsulates this DNA fragment. Further, on addition of cell, this liposome get fused or adhere to the bacterial cell or the cell where we have to transfer this DNA fragment, right? And when it adhere to the cell, further it get fused with the plasma membrane and releases this DNA to the cell, and then it goes to the nucleus, and here it is forming the recombinant DNA. So this is what the liposome is carrying the particular DNA fragment to the another cell. So that's why it is very efficient technique. It is used for transfer of genes to bacterial, animals, and plant cell. So this is what about the lipofaction. The next one is direct transfer DNA. So possible to transfer this DNA into the cell nucleus. It is very direct. We are transferring the DNA to the cell. Now the techniques involved here: microinjection, generally used for cultured cell, and it is useful to introduce DNA into the large cell, where we have a large cell like oocytes, eggs, the cells of early embryos. That time we can go for microinjection, right? If we have it at very smaller scale or very smaller level, that time we can go for the previous 5 methods, right? Transformation, transduction, conjugation, electroporation, or lipofaction. But when we have a large cells, like we can transfer the or introduce the DNA to the large cells, that time we can go for the direct transfer of DNA. So the microinjection and particle bombardment, these are the two techniques used for direct transfer of DNA. Now, what is transfection? This term is specifically associated with the transfer of DNA into eukaryotic cells by physical or chemical means. Okay, so that the transfer of DNA to the eukaryotic cell, okay, is called as the transfection. This term is specifically for transfer of DNA to the eukaryotic cell. Up till now, we have seen the transfer that in case of transformation or conjugation, the transfer is from bacteria to bacteria. So it is from prokaryotic to prokaryotic. Okay, but in case of transfection, the transfer of DNA into the eukaryotic cell. So this is all about the 6 different methods of genetic transfer. Now, what are the strategies involved in the gene cloning? Gene clone is what? Clone is nothing but the colony, right? So if we are preparing so many cells by single cell, so it is a copy of, we can say, a single cell. So that is called as a cloning. Now the clone is a group of organisms or cells or molecules or other objects arising from a single individual, right? So we used to say it, or used to say it as a clone or a colony. Now, gene cloning strategies in relation to that particular recombinant DNA technology. So there are so many aspects associated with this. Now, what are the aspects are there? Like as we have seen, the first generation of fragment is there. So that we have studied restriction endonucleases are there, which cuts the fragment of DNA. Further, the DNA ligase was there. There's all molecular tools are involved in this strategies. So the first step is the generation of DNA fragment. So the generation of DNA fragments by restriction endonuclease digestion, or by DNA synthesis, that is replication, or by PCR, that is polymerase chain reaction, or chemical synthesis. So we can generate the DNA fragments, which is the first step for the gene cloning, right, or recombinant DNA technology. Then insertion of it into the cloning vectors. We have seen the cloning vectors earlier. So we have seen plasmids, we have seen bacteriophages, then again phasmids were there, cosmids were there. So this all are the vectors which carry this DNA fragments. Now after insertion, that the ligation of the blunt ends were there, cohesive ends were there, homopolymer telling were there, and the linker molecules were there. So we'll get in detail that the homopolymer telling, what is the detailing of homopolymer telling, and what is the linker molecule. You can refer the particular reference I have suggested, where you'll get the diagrammatic presentation of homopolymer telling and linker molecule. How this linker molecules are actually linking two different DNA fragments with each other from the cohesive end, right? Or this is nothing but the homopolymer telling. Once this, with the help of this, that it introduced into the host cell. After introduction to the host cell, either it goes through the transformation, or the different ways where there, transformation, transfection, or transduction. And the last is selection or screening of the recipient cell with the recombinant DNA. So this is again done by that the screened recipient cell, or the recipient cell is having that particular recombinant DNA or not. So that is carried out by PCR, that is polymerase chain reaction. Then immunochemical methods are there, protein-protein interaction methods are there, functional complementation, hybridization. So these are the different methods used to screen the recipient cell as it is having the recombinant DNA or not. So this is what all about the gene cloning strategies. So the reference for this is again Satyanarayan. Refer it. If you find any query, you can ask me, right? So thank you so much. Here we are ending with the third session. Okay.